On today's GARNet Research Roundup, I'm absolutely delighted to be joined by Dr. Antony Dodd from the University of Bristol. And we're going to be talking about a paper just published in Plant Physiology, the title of which is The Energy Signaling Hub SNRK1 is Important for Sucrose-Induced Hypocotyl Elongation. So thanks very much, Antony, for joining us today. And so this paper has a lot of measurements of hypocotyl elongation in it. So it'd be great if you could give us the key points that you found within the paper.
Thanks, Gary. So this paper is a study about how plants measure and respond to sugars, and how those sugars regulate plant growth and plant signalling, plant development. So plants have a number of different mechanisms that they use to measure sugar and energy availability. One quite well-known example is the enzyme hexokinase, which is thought to be a sensor of the amount of glucose that's present in the cells. Another example that's becoming quite fashionable at the moment is a protein called target of rapamycin, or TOR, which is thought to be a kind of integrator of nutrient and energy signaling. And another one that we focused on in this study is an enzyme called SNF-related kinase homolog, which is like SNRK1. So SNRK1 is an evolutionary conserved kinase that seems to function as an energy signaling hub in plant cells. And the activity of SNRK1 as a protein kinase, it is involved in the phosphorylation of both metabolic enzymes to adjust their activity, and also in the phosphorylation of transcription factor proteins that leads to cascades of changes in gene expression in response to energy status. So SNRK1 is thought to become active under conditions of low energy availability. For example, starvation conditions. And so it's under these low energy conditions that SNRK1 becomes active, phosphorylates proteins, and causes some changes in the balance of catabolism and anabolism within plant cells. We were interested in our study particularly in how SNRK1 might be regulating plant development, and we used the assay of hypocotyl elongation in Arabidopsis seedlings as a tool to study development and growth. So it's a good model system to study the integration of different signals that control plant growth. So if you grow Arabidopsis on a petri dish and you put sugar in the media, under some conditions the hypocotyls will elongate more in the presence of sugars than in the absence of sugars. And so this provided us with a nice assay to study sugar signalling in relation to development. Focusing back on the SNRK1 sugar sensor, so SNRK1's a multi-protein complex that consists of a number of different subunits, and the KIN10 subunit is the catalytic subunit of SNRK1 that's really important for its activity. So we took seedlings that were overexpressing KIN10, and then we asked, well, what happens to hypocotyl elongation when we add sugar to these plants? And what we found was that under conditions where sugar causes hypocotyl elongation, the KIN10 overexpressors didn't respond to sugar when we added it, indicating that it might have a role in hypocotyl elongation. So the way that SNRK1 signals is that it uses a signaling sugar called trehalose 6-phosphate, which is a sugar that's present at very low abundance in plant cells, and is thought to function as a signaling intermediate that provides the cells with information about the availability of sugar and other metabolites. SNRK1 activity is repressed by trehalose 6-phosphate, whereas under conditions of starvation, there's not trehalose 6-phosphate present, and SNRK1 becomes active. So we also took some telling mutants of trehalose 6-phosphate synthase that were originally developed in Ian Graham's lab. And these have lower concentrations of trehalose 6-phosphate in the cells compared with wild-type plants. And we asked what happened in these mutants also when we add sugar. We said, well, what happens to hypocotyl elongation? And we found that these mutants were also relatively insensitive to sugar in terms of hypocotyl elongation.
Okay, so let me stop you there, Anthony. Also, you've set the scene beautifully here. So you said that there's these phenotypes with the KIN10 overexpression, and this cellulose synthase mutant. So this doesn't happen in all light conditions, and that's the key part of the paper. So can you explain what happens in different light conditions?
Yeah, what we found, which was really interesting, was that under conditions of longer photo periods, when you add sugar, the hypocotyls don't elongate, whereas under shorter photo periods, when you add sugar, the hypocotyls do elongate. Okay. And we wondered why this might be, because one possibility was that the plants were sensing the photo period directly through a photo period sensing pathway, and that information was being integrated with the sugar signals to control hypocotyl elongation. Yeah. Another possibility is that it related to the amount of photosynthesis that was possible during the day. So under a long day, more photosynthesis is possible because the plants are exposed to light for longer, whereas on a short day, less photosynthesis is possible. And we tested both of these ideas. And what we concluded, as is quite often the case in biology, was, well, it's a bit of both. Right. And we found that under longer photo periods, such as those that the plants might typically be exposed to between, say, 16 and 8 hours day length, under those kind of photo periods, photo period sensing and signalling seemed to be important. Okay. And in addition to that, daily integrated photosynthesis seemed to contribute to the length of the hypocotyls. Okay. Whereas under much shorter days, it was entirely down to the amount of photosynthesis that was possible. So what we concluded from that was that the sensitivity of the seedlings in terms of their development, their sensitivity to sugar, depended also on the photo period. Okay. I guess that's quite interesting because elongating seedlings in a field, depending on the season when they germinate, this might affect how well they develop and how well they establish. Yeah,
and intuitively it kind of makes sense that if you have less time to produce sugars from photosynthesis, then adding exogenous sugars as you do would have a greater effect on elongation. So there's been many studies on hypocotyl elongation over the past years, and you integrate a couple of other signaling molecules into the work. So can you explain a little bit about that as well?
Yeah, so hypocotyl elongation is driven by phytohormone signals, and there's a very long history of really elegant work in that area. And there's been papers in the past that have identified possible roles for Auxin in sugar-induced hypocotyl elongation, and also possible roles for gibberellin signalling. Okay. And there's some work also suggesting that racemose steroids might also be involved. Right. We focused in our work on Auxin and GA signalling. Okay. And one of the things that we wanted to know was whether Auxin signals might be coupling SNRK1 mediated sugar sensing with the cellular processes that lead to elongation growth. Okay. And to do this, we examined, there's a group of enzymes called expansins, and one of the things they do is they loosen cell walls so that cells can expand. Yeah, we examined a couple of these genes encoding these expansins that are auxin responsive expansin genes. Okay. And what we found was, in particular, there's one called expansin 11, which is it's regulated by auxin, but it's also induced by sugars. But in the KIN10 overexpressors, it's not induced by sugars. Right. So we suggest that expansin could form one of the links between SNRK1 mediated sugar signaling and hypocotyl elongation.
I see. Excellent. Okay, so just as a final question, as you said, a lot of this, all of the work in this paper is on Arabidopsis seedlings, but we have to think a little more about translation and more applied work. So, this work also links really nicely to some previous work undertaken by people in Rothamsted, where they looked at the effect of cellulose 6-phosphate as well. So, can you see a link between your work here and the work that they've done in Rothamsted and kind of the agronomic importance of this work ultimately?
Yeah, absolutely. I mean, there's a very nice study from the groups in Rothamsted showing that some chemicals that interfere with trehalose-6-phosphate signalling can have quite profound effects on agronomic traits found in cereals. And this suggests that manipulation of this sugar signalling pathway could provide a really nice way to adjust plant performance under field conditions. You know, you could, under certain seasons, it might be possible to manipulate what your crops are doing by spraying them with something that targets this pathway. I mean, our study was making a link between phytohormone signalling and the trehalose-6-phosphate metabolic processes. So it's possible that our study could provide one explanation for some of the phenotypes that were seen in the work done at Rothamsted. But also because our work was done in seedlings that were elongating, it gives us some information about how crops might respond if they were sprayed at a very early stage with the chemicals that are being examined in Rothamsted. So we're finding that if you manipulate trehalose-6-phosphate in elongating signalling in elongating seedlings, those seedlings will grow at a different rate. So possibly if a farmer wanted to manipulate the rate at which their seedlings become established, it might provide one way to do so, to accelerate or slow down the establishment of crop seedlings, depending on the weather conditions.
Absolutely, and you can imagine having a greater molecular understanding of what's going on then ultimately, if transgenics are in an option down the road, then this could be something to develop in future, certainly.
Yeah, absolutely.
Okay, so thank you very much, Antony, for chatting to us today. And in our previous talk, you said that there's a few other outputs soon to come out, so we'll look forward to hearing about those. Maybe we can chat about those in the future. But for now, thanks very much for discussing your work with us today.
It's been a pleasure.